Thank you. This is the first time that we've presented Opsis publicly at this meeting, or generally speaking. The company's been a little bit under the radar for the past few years, but really the company and the focus of it, myself as a co-founder, Dave Gamm, and actually Emil Neusser, who's now with BlueRock, is very similar to some of the companies in the space that are seeking to directly repair a tissue and thereby restore tissue function. In our case, our motto is rebuilding the retina and restoring vision. So the company was founded in 2016 with clinician scientist Dave Gamm, who's well known in the field for having established methods for differentiating authentic retinal cells using an organoid-based technology at the time, which we've since adapted and improved for the purpose of the company. And this was really the basis on which we established the company. The fundamental platform we're using is an iPSC-based platform, and this is built on the technology that was developed by CDI, the spin-out of Jamie Thompson and group in Madison, which was then acquired by FUJIFILM. And so in developing this program, it was spun out from that effort and financed by FUJIFILM, and it's their first external venture. And so I think it has some important significance as it relates to development of this technology for patients. Additionally, with FUJIFILM CDI, it's a sort of built-in manufacturing partner that we have. So as we develop and optimize our method, we can then scale it to the clinic by using their capabilities. And we established early on a partnership with the National Eye Institute in which we are advancing with them a program co-developed targeting AMD, in which the retinal pigment epithelium is being replaced in those patients. That program is entering the clinic scheduled later this year. So that will be our first clinical program. And what I really wanted to focus on for this talk, however, is what makes us unique, which is this photoreceptor asset that we're developing, that being the key cell type that's needed to restore function in the retinal tissues for a variety of patients, whether it's inherited or acquired disease. So I think it's important to point out that in assembling Opsis, we wanted to recruit a really top-tier advisory team, and we succeeded in doing that. And per the last speaker, this is not a kind of feather in the cap advisory team. They're heavily involved in our development, clinical strategy planning. And so there are a number of groups on here, such as Kapil Bharti from the National Eye Institute, who's our partner for the RPE program. Steve Charles is a very well-known vitreoretinal surgeon. Jose Sahel, who's very well known in the retinal regen med space, as well as Dr. James Thompson, whom I'm sure you all know is a pioneer in the stem cell field. So the platform is shown by this sort of interesting and goofy diagram, is that we have iPSCs from which we can derive very pure retinal progenitors. Those we can then formulate into organoids for a variety of applications, but we can push them into a very consistent RPE product. I won't be talking much about that product. Additionally, we can push them into cryopreservable photoreceptor progenitors, which we call PRP. And those are cells that in vivo in our animal efficacy studies, we're seeing solid formation and maturation into the target cells that we want to repair. So the mission of the company is really targeting broadly orphan diseases like retinitis pigmentosa, which affects a fairly large number of patients, even though it is nonetheless an orphan, and broadly speaking, macular degeneration. The idea here is different than gene therapy in that we're not targeting a specific genetic defect, we're targeting an anatomical defect. And so I think it's important to point that out, that it bookends gene therapy, but it's in essence potentially a much larger market. So the pipeline, as it stands right now, is really the AMD product that's entering the clinic with the National Eye Institute later this year. The other programs that we have targeting retinitis pigmentosa, which is a photoreceptor-only treatment, as well as a combination therapy combining RPE and photoreceptor, those are in preclinical stage. And I'm really going to focus just on the middle one, the RP product. And so this product is a highly pure photoreceptor progenitor. It took us 2 years and a series of candidates to really identify the right product that has the properties that we're looking for in terms of purity, formulation, scalability, and post-transplant behavior. And so as shown here, there's some information here about how we formulate it into essentially aggregates of a certain type that it's highly pure, as shown in the 2 right panels there from flow analysis. And that without disclosing the specific genes, the majority of the cells in the preparations are retinal and photoreceptor progenitors, with the rest of them being neuron and housekeeping functionality. So we've kept this rather under the radar, but we decided recently to take it on the road to interface with pharma and investors because we identified a candidate that, when delivered into a relevant model of photoreceptor degeneration, and this is a rodent model, but it has essentially lost the entire photoreceptor layer, but has an intact RPE, we're able to recapitulate consistently and evenly across the site of delivery a population of photoreceptors that then go on to mature in about a 1-to-1 ratio into rods and cones, which is important in terms of downstream applications, as well as I'll show on the next slide. What's really quite remarkable about it is that the preparation in vivo grows outer segments, which are the structural components of photoreceptors that are needed to receive and transduce light. And so this is seen over a matter of months in this particular model, and it's very consistent and robust, as you can see from the marker here, which is in green. Additionally, another question is, well, can they integrate with the host? That's a difficult question to answer just due to the limitation of the preclinical models, but the anatomical signals are all there in terms of formation of specialized ribbon synapse machinery, postsynaptic markers, that the fact that host dendrites are reaching up into the graft. And so people often ask us, do we have functional data our animals seeing? And it's a hard question to answer, so we've been much more focused on the anatomy and ensuring that anatomically the cells are doing what they are supposed to do. So I sort of conclude that the photoreceptor program we find quite compelling, and that alone could be a very large opportunity in the orphan disease space. But our mission, as set out by the top management at FUJIFILM upon financing and for our own objective, is really to develop a product that can address the large unmet need of AMD, specifically end-stage patients that have an atrophy which needs to be repaired. And so an earlier stage, but preclinical R&D effort is around how do we adhere our photoreceptor product to the RPE that we're taking into clinic now. And so this is just sort of a teaser that we've established methods for doing this, and now we're taking those methods and this product into preclinical efficacy to evaluate the feasibility of this product. So in summary, the company is really focused on 3 cell replacement products that are very highly specified with a specific mechanism of action for cell replacement, targeting, broadly speaking, orphan disease such as retinitis pigmentosa, as well as AMD. We've established our photoreceptor lead candidate. Our RPE program is moving into the clinic, and for us at least, the anatomical data are very convincing. Thanks.